Hi everyone, I'm Devesh Shukla from Chemistry Honors. Today I'm going to present my topic, which is synthesis of alpha amino acids, ionic properties, as well as reactions. So let's go. First is let's talk about the introduction. Alpha amino acids are the organic compounds containing both an amino group and an carboxylic acid group attached to the same carbon called the alpha carbon. The second is general structure of alpha amino acid. The general structure is NH2 single bonded to CH, which is also connected to a R and which is connected to a CWH, where R represents a side chain that determines the identity and properties of the amino acid. The the method for the preparation includes the one method which is Stecker synthesis, it is a classical method for preparing alpha amino acids. It involves the reaction of aldehyde with ammonia and hydrogen cyanide to form a alpha amino nitrile upon which upon hydrolysis yields the alpha amino acid. This is a very important synthesis method. It also come in the exam for almost 5 marks. The next thing is Strecker reaction mechanism. Step 1. In the step 1, the aldehyde react with the NH3 to form an imine. The second step is HCN adds across the C double bond N, that is CN bond, forming an alpha amino nitrile. The third step is hydrolysis of nitrile group, which gives us the alpha amino acid. Now coming to the second method, which is very important, it's Gabriel phthalimide synthesis. This mechanism involves potassium phthalimide that is reacting with the alpha halocarboxylic acid to produce the protected amino acids. Hydrolysis of this releases the free alpha amino acid, which is the required result, which is the thing we are going to synthesize. So this is a very important method. We have to learn it. Now, just let us talk about the mechanism of the Gabriel phthalimide mechanism. The first step that we go is nucleophilic attack by phthalimide on alpha halo acid. We have to learn the structure of phthalimide. The second step is formation of N-phthalido derivative. This is the middle step of this reaction, which is very important. The third step is hydrolysis. We get the result in the second step, and then we have to do hydrolysis as we did in the second method, and hydrolysis will yield alpha amino acid. The third is HVZ reaction, which is Hell-Volzard-Zelinsky, which we have also studied in the class 12 as well as in the first year, second year. In this, the carboxylic acid undergoes the alpha bromination with Pr2 as well as PBr3. The resulting alpha bromo acid reacts with the ammonia to give alpha amino acid. The main thing is here alpha bromination occurs with the carboxylic acid. This is the most important step, and let us talk about. Now we are going to learn about the biological synthesis of alpha amino acids. In the biological system, transformation reactions convert the alpha keto acids into the alpha amino acids using enzymes and pyridoxal phosphate. This is in the biological synthesis. We can also write in the synthesis questions. Now we are going to talk about the ionic properties of the alpha amino acids. We already talked about the steps or as well as the reactions for the preparation of amino acid. Now we are going to talk the ionic properties. First of all, amino acids are amphoteric. Second, they ionize into deuterions at psychological pH. The third is the NH2 group accepts a proton and the CWH group donates a proton. This is one of the most important things. The deuterions have high melting points and solubility in water, and amino acids are also deuterion. Very nice. Now talking about the isoelectric point. The isoelectric point is the pH at which an amino acid exists predominantly as a zwitterion with no electric charge. Here depend on the side chain. Now we are going to talk about the anionic properties. We are going to talk about the acid-base behavior. And this at low pH, amino acid exists in the cationic form. At high pH, they exist in the anionic form. Titration curve show two or more or three pK values depending upon the type of amino acid, as alpha amino acids are of many types. Now we are going to talk about the peptide bond formation. Amino acids undergoes condensation reactions between NH2 and CWH groups to form peptide bonds. Peptide bonds releasing water, they form dipeptides, polypeptides, as well as proteins. Now, we are going to talk about ninhydrin reaction. Now, we are going to talk about the reaction of the amino acids. Amino acids react with the ninhydrin to give a deep purple color. This reaction is useful in chromatography and protein analysis. Now, the reaction of amino acids with alcohol. We know that amino acids react with alcohol to produce to produce esters in the presence of dry HCl. This is very common. We already studied in the class 12. These esters are also used in the peptide synthesis. Now, next is decarboxylation reaction. I can't pronounce it. Amino acids undergo decarboxylation to yield amines. Yeah, amino acid in the presence of CO2, they yield amines which may act as a neurotransmitters. Now, one of important reaction is a transmination reaction. That is, amino acid transfer their amino acid to alpha keto acids in the presence of PLP, forming a new amino acid. Now, oxidative deamination, that is, amino acid lose their amino group as ammonia. This reaction is important in nitrogen metabolism as well as in the synthesis of nitrogen. The next is electrophoresis properties. Under electric field, amino acids migrate based on their charge at given pH. This technique helps separate amino acids. This is a very important property of amino acid. Now we are going to conclude all the PPT, all the things we have learned, and which is alpha amino acids show diverse chemical behavior due to their multifunctional nature, their synthesis, ionic behavior, and the reaction from the basis of protein chemistry we have. And we studied the alpha amino chemistry. We not only studied the preparation of alpha amino acid, we also studied the ionic properties. We also studied the reactions of the amino acids. So thank you everyone. I'm